Hi, my name is David Freire Obregón, and I'm going to present our work entitled Towards Bi-Hemispheric Emotion Mapping through EEG, a dual-stream neural network approach. In today's presentation, I will discuss our approach to the brain response to emotional avatars challenge, where we achieve a commendable second-place finish. Our study centers on participants immersed in virtual reality environment where they mimic the facial expression of an avatar, displaying 6 primary emotions. This interaction allows us to capture intricate brain activity using sophisticated multi-channel EEG equipped with advanced dry sensors. The experiment's scale is considerable, involving 40 participants who each generated more than 700 samples across different emotional states, enable a robust and detailed analysis of EEG signals. The key metrics we focus on for evaluating our results were the accuracy of emotion recognition and the processing time needed to process these signals, with efficiency serving as the deciding factor in cases of ties. To manage and analyze this extensive data, we utilized the CodaLab platform, which facilitated our code submissions and data sharing processes. This work was conducted as part of the EEE International Conference of Automatic Face and Gesture Recognition, highlighting its significance in the field of computational neuroscience and biometric analysis. This challenge not only advanced our understanding of brain responses to emotional stimuli in virtual environments, but also pushes forward the frontiers of technology in emotion recognition. Thus, the relevance basically are these 3 points: advancing neuroscience and psychology by understanding emotional responses through EEG signals that deepen our knowledge of the neurological basis of emotion, enhancing both theoretical and applied neuroscience, also improving human-computer interaction by accurate emotion detection. We can revolutionize user experience in technologies, making interaction more intuitive and responsive. And also, finally, enhancing emotional well-being by identifying emotional states accurately. This research could lead to better mental health monitoring tools, enabling timely intervention and support tailored to individual emotional needs. Analyzing EEG signals within the VR paradigm provides valuable insights into how the brain responds to emotions stimuli in immersive environments. Existing EEG-based emotion recognition techniques tackle 2 primary challenges: feature extraction and accurate classification. EEG signals offer rich data across time frequency and time frequency domains, necessitating robust feature extraction methods. This diagram illustrates the 3 module stages for Bi-Hemispheric emotion recognition from EEG signals, which are signal preprocessing, hemispheric split, and signal classification using recurrent neural networks. Signal preprocessing. We have carried out 4 different processes here. Referencing the mastoid channels, this step is crucial as it helps to minimize the influence of extraneous noise and common artifacts by referencing the EEG signal to a more stable baseline, which typically originates from the less active area like the mastoids. Also, high-pass and low-pass filtering. Implementing these filters is essential for removing irrelevant frequency components such as slow drift and high-frequency noise. Also, filter delay removal. This step corrects any temporal distortions introduced by filtering, ensuring that the timing of the EEG signal remains accurate and consistent, which is critical for precise analysis and interpretation of the time-sensitive brain activity. And finally, frequency domain representations. Transforming the EEG data into frequency domains is important because it allows for the analysis of specific frequency bands, which are associated with different types of brain activities and states, providing deeper insights into the neural dynamics. The resulting data is segregated into left and right hemispheres for further examination. EEG data captured from electrodes situated from the left hemisphere is categorized under the left hemisphere. Conversely, data obtained from the electrodes located on the right hemisphere is attributed to the right hemisphere branch. The architecture comprises two parallel streams, the left hemisphere and the right hemisphere, each processing EEG signals from these hemispheres. Each stream starts with a one-dimension convolutional layer followed by a max pooling and a dropout regularization with a dropout rate of 0.5. The output is reshaped and fed into a long short-term memory (LSTM) layer to capture the temporal dependencies. Another dropout layer is applied to prevent overfitting before flattening the outputs. This process is repeated for the signals from both hemispheres. The processed outputs then are concatenated, and the concatenated representation is passed through a fully connected layer with a ReLU activation and L2 regularization. Finally, a softmax layer produces the probabilities for the 6 emotion classes. The dataset consists on 3 sets: training, validation, and test, each serving specific purposes. The training set comprises 20 subjects, while the validation set involves only 10 subjects. Only training and validation sets are labeled, with the test set reserved for contest organizers. The validation set accuracy for various emotion classification approaches is detailed in this slide, where the baseline accuracy set by the competition is 19.4%. Notable improvements are observed with the single branch approach achieving a 23.3% and the dual branch model reaching the highest at 28.9%. This enhancement underscores the effectiveness of the dual branch architecture by improving accuracy over baseline and the single branch method. We can also see the confusion matrix for the dual branch model shown in this confusion matrix. It offers insights into performance across 6 emotions: anger, disgust, fear, joy, sadness, and surprise, indicating the predictive accuracy for each emotion category. Despite a limited number of samples from 20 training and 10 validation subjects, the matrix highlights the model's strengths, particularly in recognizing joy and sadness with a 53.3% and a 40% of samples correctly classified. We also performed a temporal analysis where EEG inputs were divided into 8 intervals to classify each interval separately. This temporal segmentation allows for a more granular examination of the EEG signals, potentially capturing transient changes in brain activity over time. Analyzing the training and validation accuracy of the monohemispheric and bi-hemispheric approaches across the 8 intervals as depicted in the slide figure reveals intriguing patterns in the model's temporal performance. Notably, both approaches exhibit higher accuracy at the initial final intervals, J0 and J7, indicating robust learning at the beginning and end of the temporal sequences. Intervals J1 and J6 notably stand out with the validation accuracy for both approaches. In particular, interval J1 demonstrates the highest validation accuracy, highlighting the significance of early temporal dynamics in accurately discerning emotions states. For monohemispheric approach, J1 achieves a validation accuracy of 25%, while for the Bi-Hemispheric approach, it reached an even higher 28.9%. Finally, we would like to say thanks to the challenge organizers, especially to Agnieszka Dubiel, and to the conference competition chairs, to Professor Escalera and to Professor Jair Escalante. Thank you very much for your attention.